So good afternoon, students. So we are moving towards the lecture number 4, that is economic importance and losses caused by the plant diseases. Actually, that are some of the examples or the previous events which is occurs or maybe happens in the history, which causes the destructive losses due to plant diseases. Particularly in, first of all, Agrios 1988 has listed the 26 diseases that have caused severe losses in the past. In 1845, late blight of potato disease destroyed the potato crops of Ireland. In 1840, population of the Ireland is 8 million, which reduces to 4 million after the famine, means almost the half million. Almost half means if you see 8 million to 4 million, many are migrated to other countries. What happens? The late blight of potato disease caused by Phytophthora infestans. Basically, that causing the hazardous effect, the toxic effects in the development of the body of humans and animals at that time. There were no any remedy or any medication for that particular disease in 1840s or 1845. So there are so many losses occurs to the particularly in case of potato fields. Next is cereal rust causes devastating epidemic losses all over the world from 700 BC. If you see in the eras, if you see in the history, there are so many eras are given in the history. So basically the cereal rust in case of wheat and other cereals, so black rust, brown rust and the yellow rust are having the epidemic losses from too many previous eras or the history in the plant pathology. In 1867, coffee rust attacked the plantation in Sri Lanka, and in 1893, the export of coffee from Sri Lanka declined by 93%. The economic crisis forced planters to cut off the coffee plants and take the tea plantation. If you see one of the other destructive disease in case of coffee, that is coffee rust. Hamelia vastatrix is the causal organism for the coffee rust, and in 1867, at that time, Sri Lanka was the topmost exporter in the coffee all over the world. Nowadays, we can say that it's Brazil in nowadays the topmost exporter. But 1893, it reduces the coffee export of the Sri Lanka decline up till 93% due to a single disease that causing the destructive losses to the coffee beans. That's why the economy of a total country forcefully collapses due to that particular coffee rust, and the planters has forcefully to cut off the coffee plant and to take the tea plantation. So that is another example for the economic losses due to the plant diseases. In India now, in 1943, the Bengal state of India faced a serious famine which caused loss of the yield of rice due to brown spot of rice, which damaging major crop of population for several years. What happens? Helminthosporium oryzae responsible for the particular disease, that is brown spot of rice, and start from 1943. If you see in Bengal as well as Orissa state, continuously destructive losses to the rice occurs every year, which causes effective losses for the development and the yield of rice cultivators. So that is the major effect also occurs in our India in some rice areas where the damaging is more due to the brown spot of paddy or brown spot of rice. Up till 1959, half of the citrus trees in the world had been destroyed by citrus tristeza virus, shortly known as a CTV. This citrus tristeza virus multiplies systemically inside the host and causing a complete death. It's known to be the sudden death, wilting stage, or the death of the plant. So it's very destructive phase to cut out the citrus plants immediately. So that known to be the citrus tristeza virus in 1959. So it originated from the planting material. If a planting material is infected by the virus, so it may cause a sudden death. So it's a destructive effect all over the world. Panama disease of banana considered as one of the important disease in history, which causes the destruction of 4 million plants of the banana in 25 years. That is another example where the rhizome or a sucker or a planting material which is to be imported from Panama Island, which is to be exported all over the world. So Panama disease that is caused by Fusarium and responsible for the destructive losses, multiplication of the pathogen inside the host systemically, and complete destruction or complete wilting of the plant or complete death of the plant occurs. So from last 25 years, all over the world, causing the 4 million losses. The epidemic of corn leaf blight caused a loss of $1 million every year. So that is also high effective in the South African and some of the African countries. Many trees are destroyed due to citrus canker disease in 1910 to 1980. There is a reporting of that heavy destruction of the citrus canker. Nowadays, you can see the citrus canker on the lime and the lemon effectively causing the heavy losses. In India, wheat rust causes a loss of rupees 40 million annually. So wheat rust, that black stem rust, yellow rust, as well as the brown rust, yearly, annually causing the losses of 40 millions. Different smut of the sorghum are responsible for an annual loss of rupees 100 million. So all over the world. So this is about the smut of the sorghum also. Chickpea, due to Ascochyta blight, Ascochyta rabii, that is the causal organism, reported 75% losses in Rajasthan, and wilt of pigeon pea causes 5 to 10% loss every year in Uttar Pradesh in a recent year. So these are some of the effective systemic and foliage fungal organism that causing the effective losses in chickpea as well as pigeon pea. In 1973, apple scab occurred in Kashmir Valley, caused 5.4 million losses in the single season, also causes rupees 50 million loss every year in Himachal region. So if you see all over the world, there is a fire blight of apple that also caused by bacterium Erwinia amyloura, and also the apple scab, that Venturia iniqualis, that causing the destructive losses to the apple orchards. So in 1973 until now, every year in the Kashmir Valley as well as in Himachal region, 5.4 million losses occurs at that time in 1973, and every year it is causing the consecutive losses in Himachal as well as the Kashmir Valley as part of the northern India. So that is about the some of the examples related with the economic importance and losses caused by the plant diseases, which is given in the form of the historical, as well as some of the how it losses causes in terms of percentage or in terms of rupees or the millions. So that is the major factor here, which is to be asked in the examination. Now, next topic, that is plant disease diagnosis. When any plant is infected, so how we can identify, that is, it depends on its symptoms. How it realize that some any physiological or naturally foliage or external part, any change in the natural behavior or physiological behavior or development of the plant, if any changes occurs, so it identifies on the basis of symptoms. So plant disease diagnosis occurs, how we can see, so it depends on the symptoms as well as signs. So first of all, definition of the symptoms. Symptoms are the expression of the disease condition which helps in diagnosis of the disease, or symptoms is the result of interaction between the host parasite under favorable conditions. So three, we already discussed that when the three pillars in plant pathology, that is host, parasite, that is pathogen, and the favorable condition, that is environmental condition, comes close to each other to form a disease triangle. And if the disease triangle arises, so it resembles the symptoms on that particular plant. So expression of the disease condition, if any kind of underdevelopment of the plant, if any foliage changes to the blindness or some kind of genetical misbehavior in the plant. So these kind of conditions to be resembles in the symptoms. And now about the signs. Signs are the actual presence of the pathogen or its structure on the host, or signs are incidental or experimental evidences of the disease causing pathogen. what happens in the signs, in case of symptoms, we can see only the sickness of the plant, or maybe sometimes external misbehavior or physiological disorder in the case of the plant. But we don't know about the symptom, that whether it is occurs by the pathogen or whether it is occurs by any drought condition or lack of irrigation facility, because we don't know, because externally only sickness of the plant can be seen. That is known to be the symptom. But in case of sign, if you see an example of powdery mildew, if a powdery growth of the fungus occurs on the leafy surface, so it can be easily identified, that is the sign, that is perfectly identified that it is the infection of fungal organism. If you see, or if you take the example of the rust, brown rust of wheat, so a brown rusty means uridospores are developed, so it is considered as a sign. So rust, smut, maybe powdery, downy, some kind of evidences actual presence of pathogen or its structure onto the fungus, onto the plant parts, which can be seen by open eyes. So that comes under sign. And whatever the misbehavior or the expression or the physiological conditions which is to be changed, physiological conditions can be seen, so it comes under the symptoms. Can be make into clarification by you have to observe that particular leaf tissues under microscope, and then you can see them under microscope the bacterial or maybe some pathogenic infections. So then you can confirm that this plant is infected by such and such pathogen. And in case of sign, you can see a clear-cut structure of the pathogen over the surface of the body, surface of the leaf, surface of the plant, by open eyes. So this is the difference between symptoms and the signs. Okay. Now types of symptoms: hypoplasia, hyperplasia, necrosis, and teratological phenomena. Basically, hypoplasia, hypo means under, and plasia means formation or development. The symptoms due to the underdevelopment of the tissue or chlorophyll resulting in dwarfing of the chlorosis respectively, this is known as hypoplasia. Underdevelopment means any such kind of which when the plant is infected by any pathogen, so certain kind of adverse effect to cause the underdevelopment of the plant underdevelopment of the tissue, underdevelopment of the chlorophyll resulting in the dwarfing of the stage of the plant. So it is known as hypoplasia. Second type of symptom is hyperplasia or overdevelopment of the tissue. That's hyper means over, plasia means formation or development. These type of symptoms are due to abnormal overdevelopment of the tissue due to increase in size or number of cells. So when it's multiple development of the cells or the thickness of the cell, size of the cell is increases, so it comes under hypoplasia, that is known as overdevelopment of the tissue. Necrosis or death of the plant. These type of symptoms are due to destruction of affected tissues involving decay or rotting of the epidermis and vascular tissues such as wilt or diebags. Necrosis means completely when any pathogen causes systemic infection to the plant, means the vascular tissues or maybe some epidermal layer. So it causes the decaying, the wilting, the rotting of the plants or a plant part. So it comes under necrosis. And the last type of symptom, that is teratological phenomena. These type of symptoms are mostly occurs and restricted with the yield contributing part, that is maybe earhead or maybe pods or maybe balls in case of cotton. So this teratological phenomena involves the rust, smut, and ergot because they are causing infection. These diseases causing infection to the yield contributing character, that is earhead, pods, etc., and fruits. So this is about the 4 types of symptoms: hypoplasia, hyperplasia, necrosis, and the teratological phenomena. Particularly, next part of this topic is laboratory test of plant disease diagnosis. If any sample, if you want to diagnose in your laboratory, so what kind of protocol or procedure you have to follow? So this is given here: incubation of the plant material, isolation and identification of biotic plant disease causal agent, diagnostic test for identification of biotic causal agent, and diagnostic test for identification of abiotic plants disease causal agent. So these are some of the methods are given here. Incubation of the plant material, one of the first step which when getting back to the laboratory may be to place a sample of the disease tissue under condition that will allow the infection agent to grow and possibly induce sporulation. Means it is to be done in BOD incubator. If you know about this, you are in the 6th semester, so you have known about the BOD incubator, biological oxygen demand incubator, that incubation creates a certain kind of atmosphere, that is the temperature and humidity 26 plus minus 26 degrees plus minus 2. So that fluctuating temperature and the humidity created inside the incubator to develop a particular growth of the fungal pathogen or a bacterial pathogen over that planting material, then plant material. Then isolation and identification of the biotic plant disease causal agent. So isolation of the fungal usually require that pieces of the infected plant tissue to place over the various nutrient media, the organism that grow on the tissue and then isolated in the pure culture. So what you have to do after growth in the incubator, then you have to take a small bits or tissues of pieces of the infected tissues of the plant parts, and then it is to be transferred over the media, which is to be taken in the petri dish, sterilized media in the sterilized petri dish, and allow it to for the growth for 2 or 3 days, and then you can find growth of the fungal and the bacterial over that particular media, and it can be isolated as a pure culture for further studies. The diagnostic test for identification of biotic causal agent. So basically, growth characters of the biotic agent can be identified under microscopic observation to study its etiology and disease cycle. So basically what happens after growth of the isolates in the pure culture or in the media, then you can take that particular organism under microscopic observation under if unless and until if you know about the general character of that particular pathogen. So you have to for identification you have to know about the general characters of that particular pathogen, whether it is fusarium, whether it is rhizoctonia, in case of bacteria whether it is xanthomonas, pseudomonas. So this kind of identified general character you have to know, and then on that basis you can identify, or maybe you can get the conclusion that this plant part or the planting material, it is caused by that particular pathogen. So this is about the diagnostic test for identification. And diagnostic test for identification of abiotic plant disease causal agent. It is extremely important to look for abiotic factors that may be important in observed symptoms. Soil and water test may be necessary to determine pH, nutrient composition, salinity, and other factors such as pesticide residues that may induce the various symptoms. So in case of other than biotic, we have seen the abiotic factors are also responsible for plant diseases. So how we can identify the abiotic plant diseases in the laboratory? So we have to go for the checking of determination of pH of that plant sample. Means maybe extraction, after extraction we can see the pH of that particular sample. Nutritional composition we have to identify particularly in that case. Then if it is a soil, then salinity, then other factors such as pesticidal residue identification on the plant sample. So these kind of laboratory highly technical protocols procedure we have to follow, and then on that basis we can go for the identification by the abiotic plant disease causal agent. So this is about the laboratory tests which we have to follow for the plant disease diagnosis. So the next point here, in the last point here in our fourth lecture, that is commonly observed symptoms and signs. So these are some of the commonly observed symptoms and signs given here. You have to read it. If any problem arises, then you can contact me on my personal phone number. So it's a simple examples are given in case of this example of the diseases or the symptoms and the sign. First is fungal leaf spot, bacterial leaf spot, vein banding, mosaic and ring spot, leaf distortion, powdery mildew, rust and smut, cankers, fruit decay and rots, wilts, shoot dieback or blights, damping off, chemical spray or air pollutant injury, and last is soil or air chemical injury. I think this already we have covered in the third semester. That's a topic that is introductory plant pathology. So you have to read it, the last point which is to be carried out here in this topic. So we have seen today about the economic importance and losses causes by plant diseases, plant disease diagnosis, in which we have seen the definition of symptom signs, types of symptoms, hypoplasia, hyperplasia, necrosis, and teratological phenomena. Also, we have discussed about the laboratory tests These are the four procedures to be followed for to identify the pathogen in the plant disease diagnosis in case of the laboratory test. So these are the commonly of the symptoms and the sign you have to read, and if any problem arises, then contact me. Okay, in the next lecture we are moving towards our main topic, that is integrated disease management, because the name of our course title is integrated disease management. So lecture number 5 will be integrated plant disease management, its concept, advantages and importance. Okay, then see you in the next lecture. Thank you.